Hello, everyone. Welcome to the RX Hope Table Talk, session number 10, a universal approach to efficiently correct protein truncation variants of SCN2A. This session is being recorded. I'm Leah Myers, and I'm the Executive Director for the Family's SCN2A Foundation. We're so excited to be bringing this virtual educational experience to our global community. During these sessions, the families and caregivers of the patients with SCN2A will be referred to as the experts, and the presenters will be the professionals. The goal of the virtual table talk is to allow experts, us, the families, unprecedented access to professional researchers and clinicians working diligently to find a cure for our children. This is meant to be a very casual environment, and your questions and comments are encouraged as we're all here to learn from each other. To keep things running smoothly, just keep your audio on mute and use the reactions button at the bottom to raise your hand if you'd like to ask a question, or you can send it through the chat if you'd like. Towards the second half, we'll just let everyone come off mute and we'll just have a discussion. And if you haven't already, just introduce yourself in the chat. Introduce your child that has SCN2A, perhaps their name, how old they are, how they present, like do they have autism, epilepsy, or both, and where you live. So I now have the pleasure to hand this over to two of our most dedicated and passionate SCN2A researchers, Dr. Chris Ahern and Dr. Aislinn Williams. Please take it away.
All right, thank you so much, Leah. It's a real honor to be here amongst all these experts. And we're really looking forward to hearing your thoughts, and any questions are a good question. So please don't be bashful. What I'd like to do is I'm just going to give a brief run through of some of the work that we've been doing, and then Dr. Aislinn Williams, my collaborator here at the University of Iowa, will share some of her approaches, and then we'll open it up for questions, maybe 15 minutes or so. And then we can open up the table for comments and interactions as they kind of roll out. So my name is Chris Ahern. I'm a professor at the University of Iowa, the Iowa Neuroscience Institute, and the Department of Molecular Physiology here in Iowa City, Iowa. My lab for many years has been studying voltage-gated sodium channels and voltage-gated ion channels. And today I want to talk to you about an approach that we developed in the last 5 or so years that allows us to use some very specific molecules in the body called transfer RNAs that we've engineered to have the ability to rescue a specific type of mutation, these stop codons that are gene inactivating mutations that I'm sure many of the experts are well aware of. And so with that, we can get started with Aislinn. So Aislinn's going to be running the slides. Dr. Williams will run the slides, and I'll be going through them. So I'll just say next, and then we can go from there. So I think the first thing I'd like to do, if everyone can see that, Leah, does that look clear on everyone else's side? Perfect. So we changed the title just a little bit. We're calling it molecular spell checking, but it's the same concept. So the collaboration is that of my group with Dr. Aislinn Williams. Aislinn, would you like to say a little quick introduction for yourself as well in the beginning? Yeah.
Sure. So my name is Aislinn Williams. I am an assistant professor of psychiatry here at the Iowa Neuroscience Institute. In addition to the project that we will tell you about today, my lab also studies voltage-gated calcium channels because we're very interested in understanding how genetic variation in ion channels contributes to psychiatric and neurological disorders. We use transgenic mice and induced pluripotent stem cells to study those genetic variants. And the data and the information from my lab will come a little bit later in the presentation.
Great, thanks for that, Aislinn. So this is our little SCN2A team. We have a great postdoc who's been helping a lot despite the headwinds of 2020, Karina Kruth, and then go back just one slide, I just wanted to say. And then we had Tierney Grisolano, who's a really great postbac, which is a position that young researchers can have in between undergrad and when they move on. She's now in medical school. And now we have the great pleasure of working with Olivia Klein, who's another post-baccalaureate research. And so this is the team that's been building the data that we'll be able to talk to you a little bit about today. So we can just go through the next slide quickly, just kind of a declaration that I work with a small biotech named Tvard that's also developing therapies for SCN1A and SCN2A disorders. So when I think about like the basis for disease, it's really about electrical signaling. So next, and the way that we think about electrical signaling, what you're looking at here is this little squiggle on the left. We call that an action potential. That's really measuring the voltage across a brain cell, a neuron. And the time scale there is just a few milliseconds, just a few thousandths of a second. So that little change in membrane voltage across a cell really controls the excitable nature of the cell. And it's really the microscopic language of neurons and how they all speak to one another. And then when you add into the complexity of all the different neurons and the connections you have in the middle, you start to grasp all the different types of connections that can be happening in the brain, which actually kind of culminates in the human experience, which is our nervous system and how we interact with the world and how our world interacts with us. But when we think about this kind of system in my lab, we really have reduced it down to the core molecular components. And one of the things, like I said in the intro, is we're really interested in sodium channels. And those are proteins that control that spike part of what we call the action potential. So sodium channels are really what control that kind of real molecular event of action potential firing. And so when you have defective sodium channels, obviously it can lead to all sorts of neurological disorders. And so when we think about sodium channels themselves, here you can go to the next slide. We actually now have snapshots or images of what sodium channels look like. These are still microscopic machines, maybe a billionth of a meter across, incredibly small. But what they serve to do is they control the flow of sodium ions from the outside of the cell to the inside of the cell. And they do so by forming a pathway that's selective for sodium ions. If you look at the slide B in the middle there, there's a small kind of selective pore looking from the top down. That's where sodium goes through these channels and it comes into the cell. By coming into the cell, it changes the electrical properties of the cell, and that leads to the action potential. But for any given neuron, you'll have thousands of these sodium channels. And every sodium channel that opens 1 million of ions per second can pour through. So you can imagine the complexity that arises when you have sodium channels kind of, controlling electrical excitability in a neuronal system. One of the things that's very encouraging about working on sodium channels per se, is much like the calcium channels that Dr. Williams was talking about, is they are established therapeutic targets. There are many small molecules, drugs, that are known to bind and regulate the function of these channels with many therapeutic benefits. So there is a track record historically in the clinic for finding ways to treat what we would call channelopathies or disorders that are associated with defective ion channel function. But the type of defect that we really want to talk about today is the project that we're working on involves a specific genetic mutation called a premature termination codon, or a nonsense mutation. And these are mutations which inactivate the sodium channel gene itself, resulting in a loss of functional protein. So you can go to the next slide. And when we think about premature termination codons in human disease, these are changes in the genetic code. And I want to go into that a little bit in the next few slides, but I'll just go through kind of just the breadth of this problem throughout the clinic. Is that premature termination codons, they're rare, many times because they're what were called somatic mutations. They're mutations that neither of the parents had, but will surface in a child. And because they're rare, they tend to be in a different spot in a different gene every time they show up. And so they're called rare diseases, but we really like to think of them as a common cause for many types of disease. And in that way, we think about that maybe there could be a common cure for many types of disease. And even though there are different types of mutations, we like to think that maybe we could find a way to correct all of them. And when you think about the scope of the problem, there are thousands of what we call rare diseases in the world. And those are types of diseases that maybe only a few thousand people in the world may have. Of those, 10 to 40% of genetic diseases are caused by these stop codons that will surface within the reading frame of a gene and cause the protein to no longer be made. But when we kind of step back for a second, there's millions of patients that have rare diseases caused by these stop codons. And so you may hear oftentimes that, oh, this is a very rare disease and only a few people have it. And that may be true, but to a certain extent, the cause is a common issue in terms of protein inactivation. There are many different types of proteins that are inactivated. In the brain alone, PTCs in 400 different genes result in known neuronal syndromes. So many of you have probably heard of Dravet, which is inactivation of SCN2A, another sodium channel gene, or RET, CDLK5, FOXG1, SYNGAP. A lot of these diseases are caused in part or in whole through these terminating mutations, which stop the production or the function of these proteins. Another challenge that I want to just kind of touch upon briefly is my last point here is that they're they're rare, but they're a constellation of rare diseases. So there's a common cause in there. But one of the other challenges is that because each one of them is individually very different, it can be difficult for gene editing or what we hear about CRISPR and all these new fixes that could repair genetic lesions. Those would be a challenge for these because each one of the mutations is quite rare. And also other types of gene therapy may not work so easily when you have a large gene like a voltage-gated sodium channel. So they really represent a unique challenge in the clinic as well as in research to come up with a new way and some new thinking about how would we even ever go about trying to provide a repair or an eventual therapy for some of these types of mutations. So you can go to the next slide, and I kind of wanted to talk a little bit more about, to make sure that everybody's on the same page about what do we mean by these inactivation mutations that stop the function of it. And so to bring you all back to 10th grade biology, I apologize in advance if this is a traumatic experience, but in the makeup of our genes, there are 4 bases for individual compounds, A, T, G, and C. Are these 4 letters that make up the genetic code of life. And so there's all the different ways you can combine these 4 letters of life to give different words, really. So if you look at the top, you have A, T, G, C, but then if you put a string of them together, you have a whole line of nucleic acids. And that's a message. When that's in a cell, that actually means something that another protein called the ribosome can translate that into a message, which is a protein. And the first thing that it does is it breaks them into 3 codons at a time. And so you can go to the next slide or just advance. There you go. So if you take that same message and just parse it, it's almost as if you had a sentence with all the words bunched together. But if you just provided spaces where they're supposed to be, you start to see there could be words that are forming with each one of those triplet series of these nucleic acids. And then if you go one step further, that can be translated into what we would call proteins. So in this case, let's just call it cure SCN2A is what that message means. But then there's another type of codon, and that's in red there. There's 3 types of codons, and those are called stop codons. They're TGA, TAA, and TAG. And what that tells the cell to do is stop making this message. It's time to stop making this protein, and now this protein needs to go off and have its function. And this is in a normal system. But if you have a type of a mutation that inactivates this system, if you want to go to the next animation, what if you have that TGA all of a sudden in the middle of the sentence, where we have a mutation that makes this middle codon now a TGA as opposed to the end? What's happened is that the protein can no longer be made because you have a period that shows up in the middle of your sentence, basically halting the translation process. So it can no longer be made. And so when you have these inactivating mutations, you'll have part of the protein being made, and then oftentimes what is remaining is not functional, and you're only left with half of the amount of protein that you really need to have the full support of the protein that's being made. And so if you want to go to the next slide, this is just an example of SCN2A, of the many types of inactivating mutations that are known, and we're knowing more all the time because of genotyping in the clinic, we're finding that SCN2A has many of these types of mutations. And what I'm showing you here is just, again, to kind of get a sense for the clinical and therapeutic challenge. Each one of these letters is a different position in the channel, and each one of these are real patient mutations. And you can see that they're scattered really all throughout the gene. It's not as though there's a hot spot that you could kind of go after, but it's really a condition that is spread throughout the entire SCN2A sodium channel. And so, we have to find a way to come up with an approach that could maybe fix each one of these regardless of where it might be. And so you can go to the next slide. And so this is kind of one of the main problems that we're trying to go after here is, so if you look at, if each one of these black lines is a different type of gene, SCN2A could be one of them. Each one of them would have a different type of stop codon within it, causing a rare disease. So you can see that each one of these red X's on the left here are different ways to inactivate all these different genes. And one of the problems is that when you have gene editing technologies like CRISPR, the way they work is they have to grab onto the local environment where the mutation is. And so when you have all these different rare diseases and all this different positional effects of where the mutations may be, it's going to be a really big challenge to basically invent and to deploy CRISPR constructs to repair each one of these constructs. So that's something I think that we will get to, but for a while, it's going to be very difficult for those approaches to help the rare disease community. There are also some small molecules such as Aislinn and ELOX is another one of these in the same family. And these are incredibly powerful small molecules. And the benefit of those approaches is you can take them as a pill, and they promote read-through of the stop codon. They kind of break the language of DNA, and they allow the protein to be made even though there's a stop codon there. But one of the challenges is with those compounds is they put the wrong amino acid in. So if you have an arginine to stop mutation, and if you promote read-through with one of these small molecules, you'll never see arginine be put back in there. So you've changed the chemistry of the side chain, and we can talk about that a little bit more in the Q&A. But what that leads to is a channel that's not normal, and oftentimes those read-through channels can have different types of function, which will be its own challenge. And so those are going to be things that are particularly difficult for sodium channel mutations, because sodium channels and many ion channels are really intolerant of amino acid changes that would come from those therapies. So we're still hoping that those are improved so that their fidelity can always be enhanced. But again, for now, those are specific challenges to the other types of therapies that are out there for read-through of small molecules. And so what we've developed at the University of Iowa is a completely different type of approach. And what we're trying to do is to use the building blocks that are already available within many cells to engineer them subtly so they can basically decode the stop signs and make them to back into the correct amino acid that they're supposed to be. And I kind of illustrate that a little bit in the next slide. Yeah, so this is this. Oh, sorry, I missed that animation. But yeah, we can skip over that. That's okay. So basically the idea is, if we have our stop codon residing in the middle of our sentence and we only have cure, but we don't have SCN2A, we have this TGA mutation, we can take a molecule that's called a transfer RNA. So I know everyone's heard about mRNA and the mRNA vaccines. tRNA is in a way a kind of mRNA. It's a very specialized form of mRNA, and that it forms this T shape. But it also basically what it serves as is all those arrows that I put there. You could almost think of each one of those as a different type of tRNA that's in your body. And it decodes a nucleic acid sequence to a protein. And it basically is the interpreter that controls the way that DNA is turned into protein, or the way that a sodium channel is made from the blueprint of the DNA. And so we thought, well, could we take tRNAs and re-engineer them in the laboratory so they no longer recognize their codon that they're supposed to see? So for instance, the C, the U, the R, or the E here, but could we engineer them so that they now saw TGA as a message to be read? And if we express those tRNAs in cells, could we promote read-through? So you can go to the next slide. Yeah, so basically turning that red arrow into a green arrow just by expressing this specific lab generated transfer RNA that now carries the ability with it to read through that TGA and turn it back into the correct amino acid. And so what my lab has been doing for the last couple of years is expanding on this idea and building this technology to the point where we have a number of different tRNAs that we've identified that are unique laboratory generated molecules. They're forms of RNA, they're tRNAs. And we now have tRNAs with the ability to rescue every known stop codon that's a cause of human disease. And in many cases, we have multiple tRNA classes that could be used to repair many of these stop codon within any type of mutation and with any type of protein transcript. Right now, we're focusing these on sodium channels, SCN2A and SCN1A, but we hope that eventually we could expand that out to go after many different types of stop codon diseases. So we can go to the next slide. Yeah, okay, I'll just go through this quickly. Yeah, so what we basically did to identify these is we looked at all sorts of tRNA genes that are out there, and then we altered them systematically to find ones that could have this new kind of really emergent behavior. And like I was saying, we now have within the lab the different tRNAs that have the ability to repair every known stop codon. So really what we're doing now with the Williams lab and my lab are trying to develop model systems to really optimize the delivery, optimize the different types of payloads that we can use that carry these tRNAs in a way that would be therapeutically useful in terms of rescuing SCN2A channels that carry stop codons. And so what we thought would be really helpful is also for Dr. Williams to talk to a little bit about the types of models that she has in her lab. So if you just go next, Aislinn, I think yours comes up after this slide. So this is just to kind of drive home the premise of what we're doing in the lab and what we're going to try and do in terms of applying it to SCN2A. So if you have a normal full-length protein, that's the blue bar there on the bottom, you make arginine would go into that spot there and it would make that full-length function. But if you have a stop sign in the middle of the gene, what happens is you get a truncated protein. And so really what we're trying to do is to make these tRNAs and then to express them in specific cells in order to read through those. And an example of this from the lab is shown on the right, where you have what's called GFP, or a green fluorescent protein. If we put a stop codon in that protein, it no longer fluoresces because we've basically chopped it in half. But if we just co-express some of our, for instance, in this example, arginine tRNAs in that cell, these are now tRNAs that no longer recognize the arginine codon, but instead go to the UGA, that TGA, UGA stop site, and then we'll rescue that genetic lesion to a very high degree. So this is this kind of seeing is believing. This is the way this technology can work, is you can have a green fluorescent protein and knock out the function, but then we can restore it by just co-expressing this kind of restorative tRNA that suppresses that stop sign to make it. But now we have a number of challenges ahead of us. So we may have what we call the payload, but now we need to develop some cell lines. We need to get mouse models, and we need to now up our game to further develop this technology to the point where it could be useful someday down the road to repair a stop codon. So with that, Aislinn, I think I'll let you take over to talk about the lines that you're developing for this project.
Sure. So I mentioned at the beginning that my lab uses a number of different model systems to try to understand what variation in given genes does to the function of proteins and how it changes the way that cells function and the way that the brain develops. One of the ways that we do that, one of the models that we use, are induced pluripotent stem cells. And I'm guessing that most of you have probably heard of them by now, but as a review, what we can do with reprogramming technology essentially is to take what is called a differentiated somatic cell. So in my lab, we take skin punch biopsies. They're really tiny little 3 millimeter biopsies from someone's arm. And we grow fibroblasts out from that. And then we reprogram those fibroblasts into induced pluripotent stem cells. Those stem cells are very, very similar to the stem cells that we can derive from embryos, except that in this case, of course, they came from someone who was living and for whom we have an understanding of what their symptoms are like, what medications they've tried, what worked and what has not worked. And we can take those stem cells and either grow more of them or we can differentiate them. And in my lab, we do both. So this is just to show you what the cultures look like. So these are fibroblasts from skin. And you'll see they actually sort of line up in ways that sort of look like your fingertip. They look a little bit like a fingerprint. We reprogram these cells into stem cells, and this process takes about 3 weeks. And you can see that the stem cells look quite different from the fibroblasts that they came from. And this is called an induced pluripotent stem cell colony. So they tend to grow in these clusters, and that makes them easy for us to pick up and then culture in order to do further experiments. So when we take these fibroblasts and turn them into stem cells, they change their shape, they form these colonies. And then the two other very important things happen. And these are changes that you wouldn't be able to physically see with your eyes. One is called telomere lengthening. So what that means is that our chromosomes have caps on the end called telomeres, and those get shorter and shorter as you age. When we reprogram cells from a person into stem cells, their telomeres actually re-lengthen as if they are in fact getting younger. The other thing that we see is what's called an epigenetic reset. So what that means is that throughout the course of your life, your DNA will develop changes to it based on your experiences, based on your environment, based on medications you take or drugs that you use. And in the process of reprogramming, most of those epigenetic changes are removed. So again, we are getting back to what is similar to an embryonic state. What's great about that is that it actually allows us to study development from very, very early time points, which otherwise we wouldn't be able to do. So in my lab, we make stem cells from patients and from people who don't have disorders. And we can differentiate those in a number of ways. So for the project with Dr. Ahern, we will mostly be doing what's called a two-dimensional differentiation, and we will generate neurons that look roughly similar to this that you see on the right here. So this is a neuron, this is its cell body. In blue are the nuclei, that's where all the DNA is. And then you'll see these red processes growing out from this cell body, and those are the neurites. So those are the parts of the cell that reach out and connect to other cells, and those connections occur at these green spots or synapses. So in the lab, what we plan to do is to take stem cells from patients who have SCN2A syndromes. We actually have 2 lines right now from patients with SCN2A syndrome. And we're going to differentiate those stem cells into neurons and then use the tRNA approaches that Dr. Ahern described to determine whether or not we can restore the sodium channels and the sodium currents that were altered by the mutations in the SCN2A gene. This, of course, is not the only thing that we can do with stem cells. So another thing that we do in our lab is we generate organoids. And I'm showing you a picture of that here at the bottom. So we can study how the brain develops from very early stages with this neuroectoderm. The neuroectoderm is the early tissue that will form the entire brain. And we can look at early brain development with organoids. So in this example, what I'm showing you is, again, blue is nuclei. So there are a lot of cells that are sort of around this hole in the middle. And that is similar to a neural tube from early development. In red, those are the cells that are actively dividing. So you can see all of the actively dividing cells are toward the center. And as those cells stop dividing and migrate out to become neurons, which is what's labeled in green, you see that they're in a very different location. So that is similar to how the brain develops in utero, and we can regenerate that in the lab. So we do not currently have a plan to use the SCN2A stem cells that we have to study these aspects of early brain development, but I understand that you have probably had some presentations from some folks who do plan to do that. So these are the approaches that our lab, that my lab is using in collaboration with Dr. Ahern to study his tRNA technology. I believe we have a little bit of a summary.
Yeah. Yeah, so just, just to impress upon you guys, that even though stop codons are rare, they're a big challenge, but there's a big commonality. So it's a very large community of patients that are all rooting for success from many different angles. And so there's a lot of approaches that are being used in other fields and other diseases that you never know may come back to help us on SCN2A and sodium channel and channelopathies. So to keep that in mind, and just to, our contribution is we're building these tRNA systems, but we just like to thank everybody for everything they've done. Team SCN2A, all the folks and Dr. Williams' lab and Karina and Olivia, and then also Leah, absolutely are just amazing to work with, and the SCN2A Foundation, and to all you experts out there. We'll look forward to getting your questions when you're ready. Thanks.
Well, that was awesome. Thank you both so much. I know we have a few questions that have come through already. And I think I'll just start it off with one that I got from our Facebook group. People are a little confused on what a protein, like a stop codon, looks like on their report. Is it always written with a letter, a number, and then an X?
Yeah, maybe Ashley.
I'm probably a little bit better suited to take that because I'm a clinician. So that is not universal. Depending on the lab report that you get, it might say something like if the mutation is R1626 stop, it might say stop. I've seen some that say like term, like TERM. I've seen some that have a little star that implies that there's nothing that comes after that. So it's not universal, unfortunately. Possibly over time, clinical genetics labs will get more consistent with that labeling, but it is not universal. And the scientific literature is just as bad in terms of not being universal.
Yeah, and also like one of the things that can also can lead to confusion is if you have an insertion of a nucleotide, that will cause what's known as a frame shift. So like just as I was saying, there's those 3 bases that go, and that's your sentence. Imagine if you shifted everything over by 1, all your letters are off now by 1 position. And so that will cause a different kind of mutation. And often that will result also in a stop codon. But that's going to be out of frame in the reading frame of the protein. So that's its own category of mutation as well. But the work that we're doing is really for where it says a star or an X or term for termination, I'm guessing. Yeah.
Okay. Yeah, because that was another question. Would the frame shifts, would this technique work with frame shift?
Unfortunately, not in the current iteration. Yeah, but I think we're starting to learn so much about tRNAs that, maybe, who knows what we can do in terms of repairing these types of small genetic errors, inserts and deletions, and maybe someday we'll get there. Right now it's just the stops, but I think there's a lot of thinking going in by not just our group. There's lots of groups out there now working on this that could be developing things for frame shifts. I think it's within the possibilities.
Oh yeah, there's so much going on. It's such an exciting time. I see, Sean, you have a question.
Hey guys, yeah, thank you so much for the chat. Really exciting stuff. Just 2 quick questions. How specific are your tRNAs for SCN2A inappropriate stop codons? Like, will your tRNA, like let's say you design one for UAG, would that tickle every UAG in the genome and lead to read through on all those, or is it designed in such a way to drive read through specifically through SCN2A? And then a follow-up question just on delivery kind of thoughts on how you're going to get the tRNA into the body and then into the appropriate neurons.
Yeah, no, those are great questions, Sean. So the first thing about tickling the stop codons, so it's not that we have specificity for SCN2A per se, but the genome and the body protects native stop codons. So all the TAGs and UAGs and TAAs that end gene synthesis in every transcript, those are protected by a very kind of complex, and it's becoming more known. But
you have a slide about this if you want me to
Yeah,
show
we could go to the end. We do have a slide kind of showing it. Is it that the ribosomal profiling, or which one is that?
Mm-hmm. Yeah, the minimal read through.
Yeah, we could look at that slide quickly, but we've looked at, we've actually tried to measure the interactions of these tRNAs with those, what we call native stops. And what the data show is that actually those native stops are very well protected from read through, not just by our tRNA, but even a lot of small molecules, not as well as the tRNA, but so we have a good kind of therapeutic window in that the tRNAs don't have really strong interactions with those native stop sites. So that's kind of where one of the first things we did once we developed the library is like, okay, we don't want to be reading through every stop because that's going to be problematic. And so the first thing we did was we did this kind of really complex assay called ribosomal profiling to look where all the ribosomes are. And it very quickly showed that the tRNAs don't have the ability to read through those native stops. So that was the first good sign. But the second point, and that's something that still we have to look at, because as we get more in vivo, there's a chance that could be happening at a very low level. So that's, our kind of therapeutic philosophy is, and research philosophy is, to be going very carefully and looking for off-target and safety issues and anything that could happen being a new approach, as well as trying to find ways to advance it and hold it back too much. So we're really trying to weigh those two efforts to make sure that we don't miss anything as we're trying to very quickly advance this. So that's kind of what guides us. So we're always looking for off-target effects at the same time. We've also looked at encoding fidelity of the right amino acid. And so far, it looks like we have good ability to encode the right amino acid. Again, those are assays that are still being developed in different cells. But you also asked a very good question about delivery. And right now for SCN2A, the most likely delivery vehicle would be AAV, because there are some AAV in the clinic. So there is kind of a path already set out there. One of the things I didn't get a chance to really highlight too much is that tRNAs are very small. So when you hear about AAVs, you'll often hear this term called payload. It's really quite literally how big of a gene can fit inside the AAV that can be delivered into the patient. And tRNAs are very small, such to the point where we could put multiple copies of tRNAs in the payload. So we almost have a payload that's too small for a traditional AAV system. So in terms of that major lift that we have for a lot of the gene editing or gene replacement strategies, we don't have that problem with the tRNAs, because the whole package is maybe 100 bases. So we think that AAV could be a really good vehicle for the tRNA delivery. And also keep in mind, the AAV field is advancing incredibly quickly. Leah, said this is an amazing time for research, and it really is. There's, major discoveries happening all the time with AAV capsids, and that have new abilities to target more efficiently and more safely. So we're really, working in parallel with other groups that are developing delivery at the same time. But we really think that AAV would be a way to have really long-term safe delivery of this payload would be the idea. Yeah, thanks for that.
Yeah, thank you.
Yeah, and one of the other things that we are working on with, I don't know that we mention these. I know we talked about them in the pre-meeting or the pre-presentation, but we do have 2 mouse models that we have engineered to have mutations that match to known patient mutations. So we have one where the mutation occurs early in the gene and one where the mutation occurs late in the gene. And we'll be using those to test these AAV approaches to see, when we give the AAV to the mice, does it go to the right cell type? Does it rescue in the cell type that we want it to? And from there, if it turns out we need to be more specific, we can change the instructions for where the gene is expressed. We can change the capsid around that so that it only targets certain types of cells. So there's a lot of work to be done on delivery with the animal models that we've started.
Great, thank you for those answers. I think you answered a lot of other people's questions with some of those. Carla, I see you have your hand up.
Hi, yes. I was wondering if you can go over the difference between blood IPSCs versus skin IPSCs. And then also second part, if a family wanted to donate skin punches, how would they do that? What is the best route for them to do?
So I apologize. I didn't hear the beginning of the first question, which was a certain type of IPSC versus skin.
Yes, blood versus skin.
Oh, blood. Sure. The short answer is that we are not entirely sure what the difference is. As best we can tell, there is no substantial difference in terms of whether or not the stem cells that you make from blood or skin are good quality, whether or not they're going to differentiate into the cells that you want them to. So many labs just sort of pick whatever works well for them and stick with it. So in my lab, we use skin for a couple of reasons, but in no way is blood a bad choice. So skin we like because those cells continue to divide, and they divide after you freeze them and thaw them again. So they are not a permanent, indefinite source, but they work for us for years and years. So when I opened my lab in 2017, one of the first things we did was to work with a local schizophrenia researcher to use some of the skin cells that she had collected from her patients to make stem cells. And they had frozen those cells back in like 2010. So they had been in liquid nitrogen for 7 years, and we grew them up and they worked beautifully. So it's really nice to have them as a source. The other thing that's really nice about them is that depending on the type of experiment you want to do, you may not want stem cells. You may actually want to keep those markers of aging. So for example, my lab studies bipolar disorder and schizophrenia, as well as SCN2A related disorders. And so bipolar disorder and schizophrenia are disorders that occur, they start later in life. They're diagnosed in the teens and 20s, as opposed to from birth or age 2 or 3. So we have some experiments where we would prefer to take the fibroblast, the skin cell, and directly turn it into a neuron and retain all of that, all those markers of aging. So that's the preference in my lab, but immediately next door to me is a researcher who always uses blood samples, and his cells work beautifully too. The other question that you had asked was regarding if people wanted to donate, what would they do? So Dr. Ahern and I are right now actually working with a group to develop a bank where we would be able to receive those. And then also, depending on the preferences of the people who are involved, the people who are donating, make those available to other researchers as well. So right now, the National Institute of Mental Health has been a repository, and I think Leah at least has been familiar, I think, with the Rutgers group that sort of is the steward, I would say, for all of the fibroblast samples that have been donated thus far to NIMH funded labs. But you can't donate directly to them. So they are a repository for cells that are collected by other researchers. So the long and the short is that it's been actually quite difficult to do, because I'll say it this way, if you were right here in town and you said that you wanted to donate, easy peasy, you show up in my lab, we take a sample, it's done. When people are not in the same state, then we have to figure out how are we going to get tissue samples across state lines with all of the people's privacy protected, and make sure that we're following the rules of each state. So that it's not impossible, it just gets a little more complicated. So that's something that Dr. Ahern and I are working on right now, because as you know, there are a lot of families that would like to contribute both here and internationally. And we just have to make sure that we're doing it right and we're meeting all the regulatory obligations.
Yeah, that's a very good point. But it's an important thing, comment to make, Carla, because those types of repository and those types of research reagents, they would be helpful for the development of any therapy. Just like the 2 mouse models that we've developed, they're not any way specific to just our tRNA approach. They could be used for the ELOCs or for anything else too. So it can become a very powerful resource for any entity that's looking to repair stops. And I think the more SCN2A stops or sodium channel stops we can have in the repository, the better. So any sorts of therapies are immediately tailored towards those large ion channel genes. So we can get a good representative coverage of what those look like. So yeah, it's something we really are striving to do, but there are many forces that keep researchers from getting at these, these reagents that would otherwise be really valuable. But know that we're trying. There's just a lot of committees to get through, right, Aislinn?
Yes, many. And this is something that we will certainly keep the foundation aware of. So we routinely have communication with the with SCN2A families, with Leah Schuss, to make sure that when we have a new resource, when we have a new project, when we have new findings, that is disseminated as quickly as possible to the people who are the most invested. So once we get this sorted out, you will know.
Hopefully we'll have our conference in Ohio, so we'll have a whole bunch of people out in the same state that we could probably coordinate something like this. Another question though, on regards with that, a lot of families have contributed blood and possibly some skin punches through the Simon's searchlight. Is that an area that you might go into and see, hey, there might be something, and would you consider taking blood versus the skin if that was more available?
Yeah, the short answer is yes. So so Dr. Ahern and I have a funded project through Safari that actually is wrapping up soon. We had a pilot project that we started, unfortunately, like immediately before the pandemic. So we have been working with the folks at Safari to determine what samples they already have. And some of them are, like you say, there have been blood samples donated and there have been some fibroblasts donated. And if those blood samples were retained in a way where we can actually recover cells from them, then we can reprogram them. If it's just like, if you go to the doctor and they take a blood sample and they just sort of leave it all mixed together, that we cannot use. But I know that Safari has worked to determine which samples they have would be suitable for reprogramming. And then of course they have the genetic information about whether or not the person who had donated had a frame shift or a stop or a missense or something like that. So yes, we are very much in touch with them because their resources are amazing.
Thank you.
Thanks, Carla. Those are good questions because a lot of us have donated blood to Simon's and they have turned them into IPSCs and they're banked. So we definitely want those being used. I think I have a list of all the variants. If you don't have a current one, I can send that over. Anybody else have any questions that's on the call that hasn't had an opportunity yet? Feel free to unmute yourself and ask or raise your hand. Amanda, I don't mean to call you out, but I know that your son has a loss of function, and I'm wondering if it's a protein, a stop codon.
Yeah, he is loss of function, but I'm so terrible at reading the genetic report. so I can't actually answer that, but I did send it to Carla. And, you know, if he does and he's a good candidate for something, I would fly him out there if you needed someone.
That's awesome. Thank you, Carla. If you find it, just type in the variant and we can let you know.
Yeah, and also I'll just throw in there if there's something that you haven't thought of right now, I'm available. We're here. We have we talked to Leah and the foundation quite a bit, and so if anything comes up and you have a curiosity or a question about anything, you can always reach out to us separately. We like to hear from folks, and so that's really where we get our inspiration and a lot of really valuable information, to be honest. So if you're not thinking of anything right this moment, but it comes to you later when you're doing your laundry or something, then yeah, feel free to reach out again.
Yeah, and I would say, I would echo that to say that for, like I said, my lab has been studying bipolar disorder and schizophrenia for a long time. And there is nothing that is as important as hearing from the people who have the disorder, or the families of the people who have the disorder that you're studying, to really direct the work that you're doing. People who do not have those disorders, people who are outside of that community, are just making their best guess at what would be most impactful. But when you have lived experience, that really strongly impacts our hypothesis development, right? So we were talking before the call about whether or not we anticipate any of our mice having seizures. And if that mouse has a mutation that a known patient has, and that patient has a certain type of seizure, we can look for that in that mouse and then see whether or not the things that we are doing, whether it be this tRNA approach or some other approach, impact that particular issue.
That's really helpful to know. And I mean, the families, the experts here, they want to contribute. They want you to move as fast as you can. So they're willing to do whatever they can. It looks like Carla typed in the variant there, and it looks like it's a frame shift, if I'm reading that correctly.
Yeah, the frame shift, yeah, those are ones that unfortunately our approach won't work yet. There are some other approaches that are out there, though. I think what Kevin Bender is working on, and some of those other groups, so those will work on some of those other inactivating mutations. It looks like there's another one, 7CT, and that's GLIN3 stop. That's right at the beginning of the channel. That would be a stop codon that we could look at. And that one is as fascinating as the other one, the tyrosine 84X, because the tyrosine 84X still makes a functional sodium channel that's no longer regulated in the way that it needs to be. So you almost wonder if whatever the patients are showing for those ones is different from the other stops. Because again, getting at what Dr. Williams was saying, for us to know some of that eventually, right now we're doing everything objectively, but if we can help look at certain phenotypes of the systems we're looking at, it's always helpful to know how well our models reflect what they're supposed to be reflecting. We don't know if we're doing a good job of really modeling these complex diseases. You want to work in simple systems that's to start, and you want to build in complexity, but we want to make sure that we're doing a good job of modeling things. Yeah, so the other one, Q1531X, we've actually worked with that one, and we've repaired that one in cells. So that last one there, that's one that we know that we can fix once we have the delivery side worked out. So some of these, that we already have the payload, we have hope of getting those repaired. And actually, why I did that work a couple months ago, so I know that we can repair those. So again, each one that we can work on that pushes us further down the road, that's all good news. And know that any energy that goes in always helps. Yeah.
Yeah, I think it's really interesting then that this is back to the mutation of the early glutamine stop. That this is a person, I don't know if I'll mispronounce her name, Isla or Isla, but having epilepsy, which is something that you wouldn't necessarily expect if the protein were just entirely missing, if you just had half of the amount of protein that was expected. Instead, I would guess that we would see something a little bit different on a cellular level, and we can look at whether or not we can target that.
It looks like we're getting some more information on how Isla presented. So she did have seizures, but they're under control now. And that is super interesting. I wonder if that has, if that has something to do, because they're both of these patients that had these stop codons early on have seizures. So there you are. Hi. Do you want to talk?
Hi. Sorry, I've been driving to soccer practice and in the car while listening. So
multitasking like a mom. We get
yes. So Isla's neurologist thinks that the possibility that she might have been more likely to have seizures because she also has a very small, like pea-sized section of damaged tissue in the left occipital lobe due to unknown causes. Who knows if it was, hypoxia during birth or infection in utero or whatnot. But her theory was that maybe because of that damaged tissue, that seemed to be the focal point for her seizure activity. So maybe that was essentially a trigger. And if it weren't for that, she wonders if she might not have had seizures, if that would have just kind of gone on normally without them.
Sure. I mean, I think one of the great things about the advent of stem cell research is that we can actually test something like that. We can test to see whether in the absence of anything else that might have happened in the environment, were those cells more likely already to undergo seizures or not. There's also a question in the chat about when do you think we will see clinical trials? And I will shuttle that to Chris, because Chris actually works with the companies that are thinking about these things.
Well, I think at this stage, what's happening is we're trying to build delivery and work in model systems as much as possible. So these SCN2A mice, for instance, are going to be invaluable in terms of getting us closer to that. I couldn't really say where we're further along than we've ever been. I guess that's something that we know for sure. We have a number of biotech companies, not just the one that I work with at Tevard, but many other companies are interested in working on these, what we call haploinsufficiencies, where you have one of these very important channel genes or synaptic proteins that's inactivated. And so there's multiple technologies and teams being developed and invested in. And with this groundswell of activity in the mRNA and genetic engineering space, you have to hope that somebody's going to get a win on one of these fronts pretty soon. So I know that there are many groups that are wanting to move towards clinical trials in the next 5 years, for sure, if not sooner. But I think because everyone's developing different approaches, it's just as fast as science can be done at this point. So it's how many hours in the day, but know that there's a lot of passionate folks out there that are trying to move things along as quickly and as safely as possible, to make sure that when we do get to that pivotal moment, that everything has been done, to the point where we know that everything's as best as it can be.
Good answer. Thank you, Chris. Sean, you had your hand up forever. Sorry.
Oh, no worries. Just kind of curious, most of the kiddos that have truncating variants have haploinsufficiency. So like ballpark 50% SCN2A expression. Kind of what's the goal and what's reasonable to expect with your technology?
Well, what we've seen so far, and this is why it's so important to get into the model systems, but we see anywhere between like 75 and 90% recovery of the protein. So it's much higher than some of the small molecules that are used. But again, in simple systems, it works really well. And so I think that's an encouraging start. So that's why we're really excited to get into IPSCs, where there are also many instances where you do see that loss of half the current. So those model systems are kind of doing a good job of recapitulating what the phenotypes look like. And so also pulling neurons from some of these mice and looking at rescue there, we'll get a good sense, because I don't think we really know yet what's the therapeutic threshold. Is rescuing, so we have 50%, that's not enough. But if we rescued 20% on top of that, would that be enough? I think that'll be a really important part that comes out of these studies. That's the kind of information we can get at a pretty early stage, we can know that, you know, recovering a certain amount, if these mice have seizures and if they have phenotypes that we can track very easily, then we can kind of really start to tie in together, how much do we need to rescue in order to have a therapeutic benefit? And again, that's information that would be useful for anybody developing a therapy that's out there. But, you know, we don't really know yet is the honest answer, but we know that with at least the tRNA technology, we can do a pretty good job of rescuing close to wild type levels of channels. So that's a real good sign for us. And that's really what encourages us to keep pushing. And it's really about delivery in many cases right now. And that's also a good sign because there's so much energy going into the delivery space these days that, you know, it's just a matter of getting things lined up. And then we could have something that would be really useful.
Sure. Yeah, makes sense. Have you done any of the titration studies to look to see kind of clinical thresholds or?
Not yet. So, you know, we just got these founder mice like within the last month. You know, the pandemic wreaked havoc across the board. And then the IPSCs were also just getting up too. But, you know, it's teed up for a really good year, Sean. We should have a lot of that information. Probably the next time we all see each other, we'll have at least tried a lot of those experiments to know. So, I mean, I think in the next year, we'll know way more than we've known in a while. And then hopefully every year after that's going to pile on. That's kind of the hope, is that we'll get some momentum going, and it won't be just us. It'll be a lot of groups that are all pushing. Yeah.
And we do have some experiments ongoing also to see how many copies of the tRNA we would want to use. So you've got a couple different ways that you can titrate things. You could titrate the number of copies that you use, because as we mentioned, they're really tiny. That may or may not give you more of what you want. Or we can titrate the amount of virus. So we call it multiplicity of infection, but how many viral particles get into each cell. So we can moderate that as well. So all of those experiments are ongoing actually right now. I shouldn't say right now because it's 6 o'clock where we are and she is at home, but this week, our post-baccalaureate student is working on experiments to look at exactly that. So is there an amount of tRNA that is too much, that is a problem, that kind of thing.
Yeah, that's really clever. So you mentioned that you could have kind of multiple tRNAs. Do each of those have like separate promoters that drive individual expression, or how does that work?
No, that design, because the tRNA, they have a different type of kind of, you know, transcriptional machinery. They don't have a promoter per se. They have what's called a short leader sequence that allows them to be expressed. So we've used the same leader. I think you could come up with a way to use different leaders if that was helpful for the design of somehow a virus. But right now it's just kind of stacked together multiple tRNAs with different leaders. And we're looking to see, would you want a lot of a virus with a few copies of tRNA, or a little bit of a virus with a lot of copies? You know, we have those levers that we can start to control. And again, all these model systems will give us access to that information that we can really empirically test every possibility to see which one works the best.
Yeah, really, really fascinating. What are your kind of early thoughts on desirable AAVs to use? And AAV9 is already, you know, approved for, you know, pretty good spine activity and tropism, but you know, what are your thoughts on kind of next generation ones, and which ones would you kind of consider for SCN2A?
Yeah, that's a really good question. I mean, right now I know there's lots of energy and in that space, to the point where I think the next generations are going to be evolved nines, you know, where folks have systematically evolved the capsid and then look to see where they are expressed. I think that those types of studies, and I think a lot of that work isn't out yet. But what we hear on the street is that there are new AAV capsids that work actually quite well. I think Shape has done a good job of identifying some of them, and that resulted in that big deal they just had recently. That wasn't for SCN2A, it was for other neurological disorders. But I think it goes to show that folks are making progress with the AAVs and that they're proving to be safe and they're working. So I think for now, that's kind of one of the go-tos over other types of delivery for the brain. Yeah.
And I think the other point to think about here is that for everything that we know about this channel, we don't actually know which brain regions are responsible for the phenotypes in each person, and different neurons have different susceptibility to different capsids. So for NAV 1.2, for SCN2A, we know it's expressed in these frontal cortical glutamatergic neurons, so in the cortex. But it also tends to decrease there over time and become replaced by a different sodium channel. In the cerebellum granule cells, that's not true. And so we don't know if we need to really target the cortex cells or if we would need to target the cerebellum as well. And are the same viruses going to do the same amount of good in each place? I think that's something that's eventually, that will take much, much longer to figure out. I think first thing is people are just going to want to see, like, can we get the virus into the brain? Does it go into the neurons? And does it correct the problems that are happening? But the longer term question that I think is super fascinating is, you know, can we then use the results from those types of trials or from our preclinical work to understand better how the brain actually generates the symptoms that you see? Because right now it's like, well, we know there's a gene problem and we know that leads to a bunch of outcomes. And then all the stuff in the middle is kind of like, I don't really quite know why it happens. And that'll be really important work for SCN2A, especially considering that, as we've said, and you guys have all experienced, each SCN2A patient is not the same, right? They have different symptoms, they have different issues, they have different things that work for them. And that's all stuff that I think it'll be very interesting to see how all of that comes out in the data.
Thank you so much for that answer. It's really refreshing to hear people say, we don't know, but we're going to figure it out, right? I mean, that's it. And for you to talk to us on that level it's so refreshing. Thank you. I have two questions and then Carla has one. One of these was from the chat and it's about a splice site. Will this approach work for a splice site variant?
I have to look at it, but I don't think so. I don't think that this will work for splice site variants. Depending on what the alteration is in the splice site, I wouldn't think so. This would mostly be in RNA that has been successfully spliced. I don't think it would work on pre-spliced RNA, but I could look at it if they wanted to send me the report, but I don't think it would. I don't think it would. Yeah.
Okay.
Sorry. Yeah.
Oh, Nicole, do you have the variant? You can type it in here.
Yeah, because I'd have to map it out onto the sequence of the gene. So I wouldn't be able to do it right this second, but I could look and see. I'm always happy to look just to be sure, but off the top of my head, I wouldn't think that type of inactivating variant would be susceptible to this type of approach. Yeah.
Okay, that's fine. Thank you so much for answering. And Nicole, if you want to send it to me, I'm happy to forward it over. And then the second question is, what will a rescue look like? Both in the mice and then like, what do we expect to see in people?
Yeah, well, you know, we can start with, we should see more sodium current. We should see more sodium channels at the very molecular cellular level. We should see less of a process called nonsense mediated decay. That's where if you have an RNA that is carrying a stop codon, that RNA becomes degraded. So you have less message to be made. So we would expect more RNA. We should impair that nonsense mediated decay process. So we should have more RNA for the channel. We should have more of the channel and we should have more sodium current that supports function. What that looks like probably in IPS models is trains of action potentials should become normalized. I think there's a fairly good track record of loss of sodium channels. You'll see altered kind of action potential, those little units of membrane change of electric change that I showed first in the slide there. Those complex trains should become normalized in a way that we can track experimentally. And then in mice, we would hope, aside from looking for off-target or toxicology effects, is that we should see a loss of seizure activity if we restore the function. Those would be the types of things we'd be looking for. But also keep in mind, we're going to be looking for more complex behaviors as much as we can within a mouse colony. That's, getting at one of the things that we have to offer with our behavior core here at the Iowa Neuroscience Institute is that we can look at what would be, close as possible to rodent versions of human autism behaviors, and maybe that's something that Dr. Williams can comment on. But the core is really built around looking at intellectual disability and or autism types of defects that are manifested in rodent models. So they're really honed on those specific types of issues. So we would be looking at first characterizing those issues and then ameliorating them with this approach. Yeah, so the types of experiments that we're planning to do with these mice, once we have a big enough number of them, as we mentioned there, we currently only have a handful of each, but once we have a big enough number of them to actually do good statistics, there are a lot of tasks that the behavior core does for complex versions of learning, ultrasonic vocalizations in pups, because as with humans, mouse pups chatter to their moms, and those sounds are disrupted sometimes in models of autism. And we'll also be looking at other things like circadian rhythms and sleep. That was something actually was discussed with another SCN2A family member, is that sleep has been an issue for their child. And so we'll be looking at those sorts of things as well. Yeah, the vocalizations are quite amazing, that they can measure that kind of form of chirping. I think it's called Deep Squeak is the program that they use to study that. Yeah.
That is so cool. That is just mind-blowing. Thank you for answering that. Carla, you have your hand up.
Yes, so there are current possibilities of treatments like an ASO. They're predicting that it would be like multiple treatments throughout the years, and we don't know if it'll stop or you just have to keep on doing that. Do you see this as a possibility of like one and done, or possibility of repeating a treatment?
I think that, I can talk to a little bit about what we'd like to get at. We'd like to have a very rare treatment intervention system where we could, if we had good specificity for the type of cells we're going after, we'd like to have a tRNA delivery event happen and then not have to do anything for years would be the ideal scenario. And that's really what we're aiming for. We know that repeated dosing is nothing that anybody really wants to endure. And so we completely appreciate that the ideal treatment will be very rare and hopefully one and done kind of a thing. I don't know how much it's known, how long you would need SCN2A, and if there's a window where it's required during development, that then we could then, if you were to lose some later on, what would be the effects of that? Those are things we just don't know yet. So I think the main goal is just to restore it as soon and as long as possible, and to develop and to keep our delivery systems in mind with that purpose. Like I should have mentioned, Sean had brought it up with AAVs, and the reason why we think AAVs is because those tend to be giving the most durable expression these days. You'll probably hear a lot of RNA. There's other types of delivery, and those are amazing for other types of conditions. But for SCN2A in particular, it just seems that the AAV was currently probably the better choice in terms of safety and durability of expression. Again, there's always technologies coming out. So anything is possible. The tRNA can be delivered as RNA or as DNA, and that they're highly versatile. So they're not limited to any one type of delivery, which is also good. So as the delivery evolves, so can our strategies to put them in place.
Thank you.
Thank you very much. Does also have, feel free to raise your hand or take yourself off mute. Anyone else that I've talked a lot. I'll give it another minute, and then we will let everyone go early and give 15 minutes back. Any last minute questions? Okay. I'm just going to do a very quick closing then. So that was amazing. Wow. Thank you so much, Chris and Aislinn, for volunteering your time to help us understand this exciting work better. I know, Chris, you did a talk for us last year during the RX Hope virtual series, but we didn't do it in this format. And it's so nice to be able to have this discussion and ask you questions directly. It gives us so much hope for our children's future. And thank you to the families who joined us today. Your questions and comments, as always, are super insightful. And if you're like me, you're going to go home, like Chris said, maybe do your laundry, and you're going to think of questions later as you digest all of this science. We're here for you, the volunteers, the staff at the foundation. We're always here. Just reach out to us, send us an email or an instant message on Facebook, and we'll do our best to get your questions answered for you. I think there are at least 2 or 3 more table talks. Check the website for the different ones that are still out, and make sure that you register for them in advance. We're going to make the recordings available as soon as possible. Carla she'll probably do it tonight because that's just how great she is. And at the end, there'll be some links that you can click on to learn more about other things. You know, as we talk, we realize there's still just so much more work to do, and we can't do this alone. We really need your help. And one major way that you can get involved right now is through the SCN2A clinical trial readiness study. If you have not registered, you've got to do it soon. We're about to close the registration. And if you have registered, but you haven't had a chance yet to fill out all of your surveys, just do me a favor and log back in and do them maybe this weekend. Just knock them out, just set up some time and take care of it. We have industry groups and researchers already requesting this data. It's super important. So there'll be links on the recording that Carla will make available. And again, thank you, Chris and Aislinn, so much for your time. We appreciate you.
Thank you so much for your questions. Like I said, the very best work that we do comes from hearing from people with lived experience. So please, you know, if you have more questions, if you have other thoughts, let us know.
Yeah, just to echo that, thank you guys all so much for your questions and for taking the time today. We'd love to see you guys in person again soon.
Fingers crossed. Bye. Have a good night. Have a nice weekend.
Thank you.